Hello, everyone. This is going to be a review for Chapter 30, Introduction to Animals. And starting off with a roadmap, essentially what you need to know is that there was a Cambrian explosion where you see a lot of animal phyla begin to emerge from looking at a few different ways of analyzing evolutionary history and genetics. We can gain a grasp of what an animal actually is. We'll talk about key innovations and diversification of animals, which gives rise to the difference between bilaterian animals and non-bilaterian animals. So first, what is an animal? So it's in opisthoconta with fungi and choanoflagellates. It's part of a monophyletic clade. Animals are multicellular eukaryotes. They don't have cell walls, but they have extensive extracellular matrix, ECM. They're heterotrophic, motile at some point in their life cycle, and with the exception of sponges, they have neurons and muscle cells. And as we go along through these chapters, a lot of these words were vocabulary we were already going over. So as some of these words are unfamiliar and I skip over them, make sure to review them in prior chapters or in some of my prior videos. So here are some major animal phyla that the book highlights that you need to know or be familiar with. I'm sure as we go along, looking more into animals, you'll see specific species in these categories. So just take a moment, pause the video and read over these. I'm not going to go over all of them since they're pretty big. And then continuing on. So like I said, there's a few different ways that we can understand animal phyla. We can look at fossils for vestigial and transitional features. We talked about that when we were going over natural selection. So go back to that if you need to review on what those vocabulary words mean. We can do comparative morphology to understand the body plan of an animal, seeing how their structures are laid out in their body. We can compare it to ancestors or to their descendants. We can compare developmental processes. So how do structures in the embryo form? At what times in embryonic development do those structures form? What are the similarities and differences between embryos of different species? And what did development look like millions of years ago down a certain lineage? And we can also, of course, compare genomes. One key animal that the book and that this chapter highlights are sponges. They're the earliest animal on the fossil record. The first sponges occurred over 700 million years ago. They're benthic, meaning that they live in the bottom of a water source. It could be marine or freshwater. They are sessile, so they are attached to a substrate. And then in terms of how they feed, they have these choanocytes, which trap food debris. They can have many specialized cell types, and some have an epithelium, which are joined cells covering body surface, such as the epidermis. Then we can begin to talk about embryonic tissue layers, also known as germ layers. Diploblastic animals have an ectoderm and an endoderm. Triploblasts have an ectoderm, an endoderm, and a mesoderm. The ectoderm gives rise to the skin and the nervous system. The endoderm gives rise to the digestive tract lining, and the mesoderm gives rise to the circulatory system, muscle, bone, and most organs. And then we also talk about the difference between radial symmetry and bilateral symmetry. Radial symmetry means you can go at any angle on an organism, cut it in half, and there's many different planes of symmetry. And then bilateral symmetry is when you cut something down the middle or in half, and it will be symmetrical like that. Bilaterians, which are bilaterally symmetric animals, are triploblastic. Again, they have an ectoderm, an endoderm, and a mesoderm. Then we can talk about how the nervous system came to be. In the book, it goes into a ton of detail about how symmetry is related to the nervous system, how all of these different features are related. So I encourage you to skim over that. It wasn't really covered much in class. We're just talking about the general key concepts that you need to understand. Cnidarians and ctenophores are radially symmetrical and have a nerve net, but some ctenophores have ganglia, which are clusters of nerves. And the nervous system in bilaterally symmetric animals is diverse. They could have a nerve net, they could have a central nervous system, a CNS. Their nervous system could be arranged in tracts or cords, and they could have nerve clusters, aka ganglia. And because bilaterally symmetric animals tend to encounter an environment at one end, you will see that the nerve cells will be clustered at that end. And I drew a picture to better help you understand. So on the left, we have a hydra, and you can see it's not really clustered anywhere. Its nervous system is branched out into a nerve net, whereas on the right, with an earthworm, it has a central nervous system with ganglia, and then the cerebral ganglion at the front of the animal. So it has a brain at the front to process that information that it receives from the front of itself. And the evolution of the central nervous system coincided with the evolution of the head, also known as cephalization. Then we talked about a big topic in this chapter dealing with the coelom. So the basic bilaterian body shape is a tube within a tube. We have the inner tube, which is the gut with a mouth and an anus. The outer tube consists of the nervous system and the skin, and then we have the mesoderm in between these tubes, which forms muscles and organs. So what is a coelom? A coelom is a fluid-filled cavity between the tubes. Its importance is that it allows oxygen and nutrients to circulate and gives internal organs independence to move. And I don't think I can, I think I can fit this if I zoom out a bit. So, okay, imagine this is an earthworm, and if you don't believe that it's an earthworm, because it really doesn't look like one, refer to figure 30.9 in the book. I'm not an artist, but I hope that you will bear with me, and we can just go through this by color. So in orange, I've marked the components of the gut, which is derived from the endoderm. So I've got my gut, and on one end I have the mouth, and on the other end I have an anus. Pretty simple. Let me skip down to blue, which is the skin and the nervous system derived from the ectoderm. So of course, on the outside, I have the skin protecting the organism. But inside, I also have the nervous system. And towards the mouth end, I drew like a dot, which again represents the fact that most bilaterians absorb the most information from their environment from the front of themselves. Skipping over to green, that's the coelom, which is lined with the red mesoderm. The red mesoderm it makes up the muscles, the organs, which I put down here below the T and gut, and then it also makes up the coelom. So again, this picture isn't beautiful. I'm not an artist. You probably have gathered that already by my other drawings, but just go back to figure 30.9 and you'll see a more anatomically accurate depiction of an earthworm. It might be a lot easier for you to understand, but this is a really important topic to understand the different ways animals are shaped, organized, structured, you name it. And then we get to some simpler, but also still confusing pictures. So there are 3 kinds of scenarios we talk about when we are considering if an animal has a coelom or not. True coelomates on the left have a coelom completely lined with mesoderm. Then we have acoelomates in the middle. Remember, the prefix A means without. So that's an animal that does not have a coelom. And then on the right, we have a pseudocoelomate. Remember that the prefix pseudo means false, fake. We saw that prefix with pseudopodia, and it has a partially lined coelom. So I tried to draw it as best I could. Again, the book has a much better depiction of these situations. But I think this should be relatively simple. It helps if you just remember these definitions. So go ahead and take a look at that. I'm not entirely sure if we even mentioned protostomes and deuterostomes, but those definitions are there if we need them. Segmentation is important. Segmentation is the division of body parts into series of similar structures. So the main thing you have to take away from this is the difference between vertebrates and invertebrates. Vertebrates have a segmented backbone made up of vertebrae, and they are a monophyletic lineage in chordata. And this really becomes key when we talk about those different lineages of animals, but we're not quite there yet. We're just building the foundation for being able to look at animals and understanding their differences, but also their similarities. If we look at their genomes, their body plans, their morphology, their evolutionary history. So these items for diversification in class, it was kind of assumed that you are pretty familiar with them, which you should be familiar with most of them. We're just talking about the different ways that animals sense their environment, different structures that allow them to sense things. We talk about different ways that they get their food, ecological roles, movement, reproduction, and life cycles. So in the book, see 30.3 for this. You can just skim it over. A lot of the terminology should be familiar to you, and the remaining terms aren't too terrible to understand. And I'm sure we'll also go over that more later on. So some key organisms that you want to focus on for this chapter would be the non-bilaterians, which, this whole time, I don't know if I've been pronouncing bilaterians right. Do I really care? Not really, because the exam's not going to be an oral exam. So, I mean, you get the point. As long as you're reading along with me, I don't think it's going to be a problem. Anyways, so like I mentioned before, we've got sponges. They're multicellular, but they lack true tissues. Remember, tissues are more organized than multiple cells, but multiple cells, multicellularity still includes a way for cells to communicate with each other. It's just that tissues are more organized, more specialized for a certain purpose within the organism. Sponges are asymmetrical. They don't have muscles or nerves. They're benthic. Most are sessile, but their larvae have cilia. They can reproduce sexually and asexually, and they're mostly filter feeders. Ctenophora, the comb jellies, have a radial symmetry. They're diploblastic. They are planktonic, and they have cilia, which the comb jellies are the largest adult animals known to use cilia. That's pretty interesting. But they don't have stinging cells. They are predators, but they use sticky coloblasts to capture prey. Most have male and female organs and can self-fertilize, although fertilization is external, so outside the comb jelly's body. And then we have the cnidarians, which include jellyfish, corals, anemones, and hydroids. They are radially symmetrical, diploblastic, and with the cnidarians, we talk about two different forms of movement. They can be polyps, which are sessile, or they could have medusae, which are planktonic. And I don't know why this cut off, but this should say stinging cells. Remember, jellyfish are able to sting people and you can die from it, so stay away from jellyfish. They are predatory. They can move with muscle contractions, or their larvae can move with cilia. They can reproduce asexually or sexually. Sessile polyps reproduce asexually, and these include anemones and corals. In free-floating medusa, which are, the jellyfish that you see just out in the water. When you think of a jellyfish, you'll think of the medusa. Those reproduce sexually, although fertilization there is usually external, outside of the body. And that's it. I hope that this was a short review. It's a short chapter. Make sure to read ahead for chapters 31 and 32, and have a good day.